Assalamu alaikum dear viewers, I once again welcome you to my channel, and today we'll be discussing about Golgi complex. If you remember our last class, in our last class we have discussed about the endoplasmic reticulum. Okay, and if you remember the functions of rough endoplasmic reticulum, the rough endoplasmic reticulum are involved in the synthesis of proteins. Once again, let's have a look here. This is nucleus, and with this nuclear membrane, this endoplasmic reticulum is a continuous structure. It is the network of these tubular structures. And these are rough endoplasmic reticulum, or rough endoplasmic reticulum has these granular structures, or we call them as ribosomes attached. फाइन और जो है हमने रफ एंडोप्लाज्मिक रेटिकुलम के फंक्शन के बारे में कहा था कि एज दिस रफ एंडोप्लाज्मिक रेटिकुलम हैज दीज राइबोसोम्स अटैच्ड ऑन इट्स सरफेस, सो दीज राइबोसोम्स बीइंग द प्रोटीन फैक्टरीज ऑफ द सेल, this rough endoplasmic reticulum is involved in the synthesis of proteins. Fine. Now, once these proteins are synthesized by these ribosomes, what happened next to these proteins? Okay, and if you remember, I have told you that this rough endoplasmic reticulum is involved in the synthesis of such proteins which has the destination usually outside the cell. Okay, which are secreted by the cell outside, and I have given you the example of insulin. Fine. The islets of Langerhans are the cells present in the pancreas which are involved in the synthesis of this insulin, and the insulin needs to be secreted outside these cells. So such type of proteins which has destination outside the cell, which has its destination in the extracellular environment, so such type of proteins are synthesized by the rough endoplasmic reticulum. Now, once this rough endoplasmic reticulum के पास जो राइबोसोम अटैच है, once it जो है synthesizes the protein, this protein is secreted inside the lumen of endoplasmic reticulum. Suppose this is the protein here, secreted by the rough endoplasmic reticulum, secreted or synthesized by the ribosomes attached on the surface of rough endoplasmic reticulum. Fine. Now, this protein is secreted or sent inside the lumen of this endoplasmic reticulum. As we know that this endoplasmic reticulum, these tubular structures, have the hollow space inside which is called as the lumen of endoplasmic reticulum. तो जैसे ही ये राइबोसोम ये प्रोटीन बनाता है, और फिर जो ये राइबोसोम जो है जो रफ एंडोप्लाज्मिक रेटिकुलम की सरफेस पे अटैच रहता है, तो ये जो प्रोटीन बनाने के बाद इसे इस ल्यूमन में भेज देता है. ओके, बट स्टिल दिस प्रोटीन जो है ये अभी जो है प्रॉपर मॉडिफाई नहीं हुआ होता है, ये प्रॉपरली बना हुआ नहीं होता है ताकि इसे हम यूज कर सके. इट नीड्स फर्दर मॉडिफिकेशंस, और वो मॉडिफिकेशंस करने के लिए ये रफ एंडोप्लाज्मिक रेटिकुलम जो है ये इस प्रोटीन को वेसिकल्स में पैक करके इस प्रोटीन को फर्दर मॉडिफिकेशन के लिए गोल्गी एक जो है सेल ऑर्गेनेल तक भेज देता है, जिसका नाम है गोल्गी कॉम्प्लेक्स. ओके, और इसी गोल्गी कॉम्प्लेक्स के बारे में टुडे वी आर गोइंग टू डिस्कस. फाइन. सो वंस दिस प्रोटीन इज secreted inside the or sent inside the lumen of endoplasmic reticulum, this endoplasmic reticulum जो है इस प्रोटीन को पैक करता है in the vesicles, and that vesicle जो है, now it contains the protein. This protein needs further modifications, and these vesicles are called as transfer vesicles, and these are also called as the reticular vesicles. Fine. So once this ribosome, which is attached on the surface of rough endoplasmic reticulum, it synthesizes the protein. It sends this protein inside the lumen of endoplasmic reticulum. But now this endoplasmic reticulum packs this this protein in the form of vesicles, and these vesicles are called as transfer vesicles or reticular vesicles. Fine. And in the form of these vesicles, these proteins are sent from the rough endoplasmic reticulum towards a cell organelle, which is called as the Golgi complex. Fine. Now this Golgi complex was named after its discoverer, जिसका नाम था Camillo Golgi. फाइन. कैमिलो गोल्गी एक साइंटिस्ट था जिसने पहली बार इस गोल्गी apparatus को और गोल्गी कॉम्प्लेक्स को सेल के अंदर डिस्कवर किया था. तो इसी वजह से जो है इस ऑर्गेनेली को जो है नाम दिया गया गोल्गी कॉम्प्लेक्स और गोल्गी apparatus और गोल्गी बॉडीज. फाइन. नाउ दिस गोल्गी apparatus जो है ये इसमें जो है flattened membranous sac like structures होते हैं, और these flattened membranous structures are 3 to 20 in number, and these flattened sac like structures are called as cisternae. Okay, again, just like here we called these tubular structures as cisternae, here also in the Golgi complex, these flattened sac-like structures are called as cisternae. फाइन. नाउ ये सिस्टर्नी जो है ये इस गोल्गी कॉम्प्लेक्स में इनका जो नंबर है वो वैरी करता है. देयर कैन बी मिनिमम ऑफ 3 and maximum of 20 cisternae in the Golgi complex. Fine. Now the number of Golgi complex in the cell also varies. जिस सेल को ज्यादा प्रोटीन बनाने का काम होता है तो उस सेल में जो है Golgi complex का जो नंबर है वो वैरी करता है. Fine. So there is minimum of 1 Golgi complex present in the cell. So once these proteins which needs further modification, these proteins are जो है packed in these vesicles which are called as reticular vesicles or transfer vesicles. Now this reticular vesicle comes and fuses with this Golgi complex. Okay, and this Golgi complex जो है it has 2 ends. Fine. One end is facing towards the rough endoplasmic reticulum and one end is facing towards the cell membrane. Fine. And the end which is facing towards the rough endoplasmic reticulum, जो हिस्सा Golgi complex का rough endoplasmic reticulum की तरफ होता है, that part is called as cis phase. Okay, and it is also called as cis Golgi. फाइन. और जो वाला हिस्सा रफ एंडोप्लाज्मिक रेटिकुलम से अवे होता है एंड इट इज टुवर्ड्स द सेल मेम्ब्रेन, एंड दैट पार्ट इज कॉल्ड एज ट्रांसफेस और ट्रांसगोल्गी. Fine. So we have seen that Golgi complex has 2 ends. One is cis phase, which is towards the rough endoplasmic reticulum, and one is the trans phase, which is towards the cell membrane. And all these flattened structures, all these cisternae, which lies in between the cis phase and trans phase, are called as intermediate Golgi. Fine. So we have seen this Golgi complex is a set of these flattened membranous sac-like structures which are called as cisternae, and there can be 3 to 20 cisternae present in the Golgi complex, and depending upon the functions of the cells, these Golgi complex का जो number है that also varies inside the cell. Fine. Now this Golgi complex has two faces. One is the cis face, which faces towards the rough endoplasmic reticulum. It is also called as the entry face because this vesicle which is sent from rough endoplasmic reticulum, which is called as transfer or reticular vesicle, this vesicle enters the Golgi complex from this face, which is called as cis face, cis Golgi, or the entry face of the Golgi apparatus. Fine. And the other side which faces towards the cell membrane is called as transface or trans-Golgi, and it is also called as the exit face of the Golgi complex, because all the modifications are done inside this Golgi complex, and then the proteins are transported to their destinations through this phase, which is called as the transphase or exit phase. And all the cisternae which lies in between this cis phase and transphase are called as intermediate Golgi. Fine. Now, once these transfer vesicles come and fuses with the cis phase of the Golgi complex, say for example here, okay, they come and fuse with the cis phase of the Golgi complex, and the proteins जो है, they are sent inside the lumen of the Golgi complex. Fine. Now these proteins which were formed here by the ribosomes on the surface of endoplasmic reticulum, they are sent to lumen of endoplasmic reticulum, but still they are not fully modified. They are not still usable proteins. So these proteins, in order to make them usable, जो है, these proteins are sent in the form of vesicles to the Golgi complex, and these proteins are delivered inside the lumen of the Golgi complex. Fine. Now the some of the modifications are done here in the cis Golgi, और इसके बाद जो है these proteins जो है, they are sent in the form of transfer vesicles to the intermediate Golgi for the further modifications. Okay, and the enzymes, some of the yes, now these proteins are sent to the intermediate Golgi in the form of these transfer vesicles for the further modification. Okay, and this process continues like this for the further modifications until this protein reaches here to this transphase or trans-Golgi. Okay, now this protein is fully modified, and what are the modifications? For example, the glucose or carbohydrate is added to the protein to make it glycoprotein. Okay, and to some proteins lipids are added and they are made the lipoproteins. Fine. So these are the modifications which are done inside the Golgi complex. Fine. I'll repeat. Once this transfer vesicle comes out from the endoplasmic reticulum, it fuses with the cis phase of Golgi complex, and the proteins are delivered inside the cis phase, inside the lumen of the cis Golgi. Fine. Some modifications are done here, but for the further modification, जो है, this cis Golgi transports this protein to the intermediate Golgi in the form of these transfer vesicles. Okay, and this process continues and until these proteins reach the trans-Golgi or the last cisternae of the Golgi complex. Fine. Now we have these proteins ready for the transport. Okay, so these Golgi complex are not only involved in the modifications of these proteins, but they are also involved in the sorting of these materials. They are also involved in the sorting of these proteins depending upon the destination of these proteins. इन proteins को हमें कहाँ पे deliver करना है, उसकी बुनियाद पे इन proteins की sorting भी Golgi complex के अंदर होती है. Okay, and inside the Golgi complex, these proteins are packed in different vesicles depending upon the delivery or depending upon the destination of these proteins. फाइन. कौन सा प्रोटीन हमें कहां पे किस प्रोटीन की जरूरत है, उसी बुनियाद पे जो है गोल्गी कॉम्प्लेक्स में इन प्रोटीन्स को इन डिफरेंट वेसिकल्स में पैक किया जाता है. For example, some proteins are packed in the membrane vesicles. Okay, some proteins are packed in the membrane vesicles. Membrane vesicles, and these proteins are delivered to the cell membrane. इन proteins की जो है destination होती है वो cell membrane होती है. If you remember, there are proteins present in the cell membrane. For example, the integral proteins. Those integral proteins are processed, modified, and sorted, packed here in the Golgi complex, and these are packed in the form of membrane vesicles, and these membrane vesicles go and deliver these proteins to the cell membrane. Okay, and some of the proteins are packed in the form of secretory vesicles. ओके, कुछ प्रोटीन्स को जो है वेसिकल्स में पैक किया जाता है, विच आर कॉल्ड एज सिक्रीटरी वेसिकल्स. एज द नेम इंडिकेट्स, दीज टाइप ऑफ प्रोटीन्स जो है दे आर सिक्रीटेड आउटसाइड द सेल. These secretory vesicles take these proteins to the cell membrane, and by the process of exocytosis, जो ये proteins है इनको सेल के बाहर भेजा जाता है. Again, the example of the insulin, okay, which is synthesized inside the beta cells of islets of Langerhans, and these proteins, for example insulin, they are packed in the Golgi complex in the form of these secretory vesicles, and these secretory vesicles take these proteins to cell membrane, and by the process of exocytosis these proteins are transported out of the cell. Fine. And then some of the जो है proteins or some of the materials are packed in the form of transport vesicles. Transport vesicles. Okay, and these transport vesicles के अंदर जो materials होते हैं, जो proteins होते हैं, they are meant to send inside the cell. ओके, उनको सेल के अंदर, फॉर एग्जाम्पल किसी सेल ऑर्गेनेली को प्रोटीन की जरूरत है, फॉर एग्जाम्पल माइटोकॉन्ड्रिया तक हमें कोई प्रोटीन पहुंचाना है, तो दैट प्रोटीन टू एनी सेल ऑर्गेनेली inside the cell, that protein is delivered through these transport vesicles. Okay, and say for example we have to deliver the digestive enzymes to the lysosomes. Okay, and these digestive enzymes are delivered to the lysosomes. These are also packed and sorted inside the Golgi complex, and these जो है digestive enzymes are delivered to the lysosomes through these vesicles, which are called as transport vesicles. Fine. So now we have seen that reticular vesicles are coming from endoplasmic reticulum, rough endoplasmic reticulum, and these reticular vesicles are containing the proteins. These proteins need further modification, further sorting, processing, packaging, and for all these things, these reticular vesicles are taking these proteins from rough endoplasmic reticulum to the Golgi complex. This Golgi complex has the two faces. phases, cis phase and trans phase, and cis phase is called as the entry phase because these reticular vesicles come and deliver these proteins to Golgi complex through this cis phase. Okay, and this reticular vesicle come and fuses with the Golgi complex or cis phase of the Golgi complex and deliver the proteins inside the lumen of the Golgi complex. Fine, and this lumen जो है, this cis phase or cis Golgi now transports these vesicles through these transfer vesicles, and these transfer vesicles taking these proteins for the further modifications towards the intermediate Golgi, and the process continues till the protein is completely modified, sorted and packed inside the Golgi complex, and it reaches the trans phase of the Golgi complex or the trans Golgi. Fine, now this protein जो है, it is ready for the transport, it is ready for the delivery, and depending upon the destination, these proteins are packed in different vesicles. If the protein has to be sent outside the cell, they are packed in membrane vesicles. If the protein is sent to the cell membrane, for example integral proteins, that those proteins are जो है packed in the membrane vesicles, and if the protein has to be delivered to any other cell organelle, these type of proteins or enzymes are packed inside the transport vesicles. So that was all about the basics of Golgi complex. I hope I was able to make you understand this, these basics of Golgi complex. And if you have any suggestion, all your suggestions are most welcome in the comment section. Please like and share this video. If you have not subscribed to my channel yet, please subscribe and press the bell icon. Thank you.